The reason Unit 10 is called cell cycling is because there are different types of cell cycling. Video 10.7 is on a special type of cell cycling called meiosis, a type of cell cycling that's used in sexual reproduction. This is a big video that accomplishes meiosis goals, but also will have to engage our mitosis brains to compare these two processes, mitosis and meiosis. Pause the video to take a closer look at the goals for this video. Before getting into the nitty-gritty of meiosis, we have to add some depth to our knowledge of reproduction. Meiosis is used for sexual reproduction, but let's tackle asexual reproduction first. Asexual reproduction is a reproductive strategy that makes genetically identical offspring. There are a ton of organisms that reproduce this way. Here you see bacteria reproducing asexually by binary fission, and yeast and hydra reproducing asexually by budding. Protists like amoebas or paramecia reproduce just by doing mitosis, and more complex organisms like some lizards and fish and amphibians and insects can reproduce asexually by a process called parthenogenesis. In parthenogenesis, the female can make eggs that don't need to be fertilized to make babies. Anyway, there are some advantages and disadvantages to asexual reproduction. Write this down. For advantages, if the individual that's reproducing asexually has fit traits, then asexual reproduction is okay. That individual that's reproducing asexually is surviving in the current environment, so all of its offspring should be able to survive too. But then what if the environment changes? In that case, it's advantageous to have genetically different offspring. If the environment changes and all of the offspring have the same traits, then there's a huge risk that all of the offspring could die if they're identical. That is bad fitness. So then, sexual reproduction is a strategy that makes genetically different offspring. Sex cells are gametes from two different organisms joined together to make the offspring. But simply joining together cells wouldn't work. As you know, humans have 46 chromosomes. If a sperm and an egg had 46 chromosomes each and then fused together, the next generation would have 92 chromosomes. If that generation had sperm and eggs with 92 chromosomes each, then the next generation would have 184 chromosomes, and, well, things would just get weird. So there needs to be another way to make sperm and egg cells. That other way is meiosis. Meiosis is nature's way of decreasing or cutting in half the amount of DNA in sperm and egg cells. Sometimes meiosis is called a reduction division because the amount of chromosomes decreases by half. This diagram over here shows how it all works. All people start as one cell called a zygote. A zygote has 46 chromosomes. To make a whole organism, the zygote divides by mitosis and makes an adult organism. Only the cells in the testes and ovary that make sperm and egg cells would do meiosis. These specialized cells do meiosis to reduce the chromosome number to 23. Then, fertilization is the process where the sperm and egg join together to make a new organism. Sperm and egg and the new organism. In humans, a sperm with 23 chromosomes joins an egg with 23 chromosomes to undergo fertilization. The resulting zygote will have 46 chromosomes, and this makes a cycle, because the zygote cell then just divides and divides and divides and divides by mitosis to become a fully grown adult. Some of those cells undergo meiosis to become sperm and egg, and the cycle goes on and on. Now, there are some terms on this diagram that we need to master, and those terms are diploid and haploid. The cells we've been talking about so far in this unit are diploid. Diploid cells are the cells with 2 of every chromosome, the maternal chromosome and the paternal chromosome. In humans, this means 2 chromosome 1s, 1 each from mom and dad, 2 chromosome 2s, and so on until you get 46 total chromosomes. Somatic cells have 46 chromosomes. Somatic cells are pretty much every cell in the body except sperm and egg cells. Haploid cells have half the amount of DNA, or only 1 of each chromosome. Gametes, or sex cells, are haploid cells. Gametes can have either the maternal, mom, or paternal, dad, version of each chromosome. So a gamete could have the mom version of all the even chromosomes and the dad version of all the odd chromosomes, or any possible combination in between. From a cell biology perspective, meiosis looks a lot like mitosis, so having mastered mitosis will help you a lot here. Scientists think that mitosis evolved first, but then meiosis was an evolutionary kind of spin-off of mitosis. Understanding meiosis really depends on you remembering these terms: chromosomes, chromatids, and homologs. So for a quick review, let's check out this cell. This cell has 6 chromosomes: 1, 2, 3, 4, 5, 6. Each chromosome has 2 chromatids, 1 on the right and 1 on the left. In this picture, there are 3 homologous pairs. Let's say that red chromosomes came from the dad, so all of these came from the dad, and blue chromosomes come from the mom, so all of these came from the mom. So there's a homologous pair of chromosome 1s, the biggest chromosomes, a homologous pair of chromosome 2s, and a homologous pair of chromosome 3s. At a quick glance, the process of meiosis kind of looks like mitosis twice. The first part of meiosis is called meiosis 1, and the second part of meiosis is called meiosis 2. There's a critical difference, however. Remember our mnemonic of IP, MAT, C? Here are those phases in meiosis 1. Interphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. In meiosis 2, however, there's no interphase. There's no interphase 2 on here. This means that after the first meiosis, the cell won't get any bigger because there's no G1 and G2 phase, and more importantly, the DNA won't replicate a second time. Now, when we see meiosis as a series of stories, we can fold in the same stories as mitosis. The reason that the nuclear envelope, DNA structure, and DNA location change are all for similar reasons in mitosis and meiosis. What's new about meiosis, however, is keeping a closer eye on the homologous pairs of chromosomes, keeping a closer eye on chromatids, and keeping an eye on the diversity of the gametes. Let me tell you what I mean. Let's check out that same diagram as before, but look at the homologous pairs of chromosomes. Remember, we're going to call the mom version as the blue one and the dad version as the red one. So in this cell, there are 4 chromosomes, a chromosome 1 from the dad, a chromosome 1 from the mom, a chromosome 2 from the dad, and a chromosome 2 from the mom. In metaphase 1, over here, the homologous chromosomes line up together along the metaphase plate. This is different than in mitosis. In mitosis, there was a single file line of chromosomes at the metaphase plate. Meiosis kind of uses the buddy system with a double file line at the metaphase plate of meiosis 1. This means that at anaphase 1, what's separating is the homologous chromosomes. Here's the red homologous chromosome 1 and the blue homologous chromosome 2 going towards this pole, and the blue chromosome 1, red chromosome 2 going towards the opposite pole at anaphase. Now what this means is that at the end of the first meiosis, the resulting cells get either the maternal homolog or the paternal homolog. To state that simply, meiosis 1 divides the homologous chromosomes into different cells. Again, the first meiotic division separates homologous chromosomes so that the two daughter cells get either the maternal or the paternal double-stranded chromosome. Now let's reboot the story, but look at the story in terms of chromatids. In the meiosis 1 division, the sister chromatids stay together. So you can see at the end of meiosis 1 how the sister chromatids are still together in double-stranded chromosomes. In meiosis 2, however, each double-stranded chromosome lines up at the metaphase plate in single file. Meiosis 2 looks like mitosis. During anaphase 2, the sister chromatids split and move to either centriole at opposite sides. What this means is that at the end of meiosis 2, each cell ends up with a single-stranded chromosome of either the maternal or paternal chromosome. In other words, meiosis 2 separates sister chromatids from one another. So meiosis is different because meiosis is two divisions, but the thing to be aware of is that meiosis has only one replication of DNA. Normally, cells have two of each chromosome. During S phase of interphase 1, the DNA is copied. Then, the first meiotic division separates the homologous chromosomes. The first meiotic division changes the diploid cells into haploid cells. Because the number of chromosomes is halved by the end of the first division, meiosis 1 is often called a reductive division. Meiosis 2 follows, but without another DNA replication. Because meiosis 2 divides the double-stranded into single-stranded chromosomes, the overall number of chromosomes doesn't change. The cell started as haploid at the beginning of meiosis 2 and ends at haploid. Meiosis 2 just changes the haploid cells with double-stranded chromosomes into haploid cells with single-stranded chromosomes. Think about how difficult sexual reproduction is. First, you have to put yourself out there to find a mate. Finding a mate often means exposing yourself to predators out in the wild. There's often intersexual selection and intrasexual selection, which uses up energy and may even cause injury. Now, we've added the extra step of meiosis to the mix, so there must be a point to all of this trouble. Well, the huge advantage of sexual reproduction is creating offspring with genetic diversity or genetic variation. If there's a disadvantage to the offspring, well, at least it's probably not to all of the offspring because of that diversity. Meiosis and fertilization create diversity in three different ways, with a process called homologous recombination, with a process called independent assortment, and with random fertilization. A more simple name for homologous recombination is crossing over. Crossing over happens during prophase 1. During prophase 1, the homologous pairs, they pair up. And during this homologous pairing of prophase, the pieces of chromosome arms that are next to each other physically cross over and swap equal pieces of DNA. You could see this process right here. At the start, each double-stranded chromosome is all blue or all red. When the chromosomes cross over, though, they swap pieces of the chromosome arms that are near each other. So now, this one has blue and red, and this one has red with a little bit of blue. At the end of meiosis, as you can see down here, now there's at least 4 different versions of the chromosome. All blue, all red, mostly blue with a little bit of red, and mostly red with a little bit of blue. Here's a zoomed in picture. The process of homologs pairing up is called synapses, which is happening here. Then, homologs swap equal pieces of DNA. That's the part that's crossing over. Then, you get 4 different versions of each chromosome. All green, mostly green with a little bit of purple, mostly purple with a little bit of green, and all purple. The second mechanism for making genetic diversity is called independent assortment. What independent assortment means is that the way chromosome 1 separates, in this case with blue on the top and red on the bottom, is independent of the way that chromosome 2 separates, in this case with red on the top and blue on the bottom. In other words, chromosome 2 could have just as easily been with blue on the top and red on the bottom. Chromosome 2 isn't based on what chromosome 1 did. With just these 2 chromosomes, you can end up with cells that have 2 reds, 2 blues, a red chromosome 1 and a blue chromosome 2, or a red chromosome 2 and a blue chromosome 1. So 4 different combinations. But now, imagine you're dealing with 23 different independently assorting chromosomes. What that means is that there's 2 to the 23rd possibilities. That's over 8 million possible combinations. Now that's some genetic diversity. So just by this process, you can make 8 million different sperm and 8 million different eggs. So the possibilities seem endless, but diversity isn't done. Let's add reproduction to the mix. So to kind of simplify, let's think about independent assortment, right? So independent assortment makes over 8 million different sperm and 8 million different eggs. The chances of a genetically identical sperm and a genetically identical egg meeting to make a genetically identical offspring are less than 1 in 64 million. But then, if you consider adding crossing over to the mix, there's genetic variation galore. The processes of meiosis combined with fertilization really mix things up. Now, if you put mitosis and meiosis side by side, you'll see some similarities and some differences. So both mitosis and meiosis are types of cell cycling or cell division. Both processes use similar mechanisms with mitotic spindles that you can see here, nuclear membranes changing and packing up chromosomes. But the goals are different. The goal of mitosis is like a cell copy machine, to make identical copies of cells for growing or repairing or regenerating tissue. The goal of meiosis is to make a diverse set of gametes, so that at the end, the cells have different genetic material than at the start, and the cells have half the genetic material as the cells at the start. During the first division, meiosis halves the number of chromosomes from diploid to haploid, and during the second division, the chromosomes go from being double-stranded to single-stranded. The end result is cells with half the number of chromosomes and chromosomes that are different. So let's recap what we've learned here. First, we learned about reproduction. Asexual reproduction creates genetically identical offspring, which are really good if the environment is stable and you know that all of the offspring are going to survive with those successful traits. Sexual reproduction, however, has the advantage of creating genetically diverse offspring, which are advantageous in changing environments. So maybe not all of the offspring will survive, but not all of the offspring will die either. Meiosis is a process that uses two-cell divisions to reduce the amount of DNA and create genetically diverse gametic sex cells used for sexual reproduction. The process of meiosis has mechanisms built in to create genetic diversity. Those mechanisms are homologous recombination and independent assortment. Those, along with random fertilization, create genetically diverse offspring, which are really good in a changing environment.